Next speaker today is Dr. Scott Selleck. Dr. Scott Selleck is the professor and department head of biochemistry and molecular biology. His research focuses on understanding nervous system development. His lab uses various genomics, genetics, molecular, and cellular methods in the model organism fruit fly. Most recently, using human genetics, his lab has identified regions of DNA that can contribute to developmental abnormalities like autism. And today he's going to talk to us about genomic dynamics and instability in autism. So everybody, please your hands together for Dr. Selleck.
Good afternoon, or good morning. Thanks for this opportunity. I'm going to start with, this was my formal title, but in light of the type of presentation, I'm going to switch it a bit, and I'm going to tell you something that we all know, but I'm going to tell you some details about it that you may not know. And that is, if this works, which it doesn't, of course. Humans are unstable. You know this is true. You've interacted with them. The question is how and why. And today I'm going to limit my discussion of instability in terms of the genome. Now, what we're really trying to understand in human genomics, in large measure, is this question. What is the source of the variation between us? And there's a lot of it. In terms of the characteristics we see all the time, whether that be our facial expressions or our ability to do physics. There's also a lot of variation with regard to disease susceptibility. So one of the major challenges we have in modern human biology is to figure out what is the molecular basis of this variation and can we predict it. And with regard to disease, for example, if we can predict it, can we do something about it? Can we actually identify people that are susceptible to disease and do something before that disease emerges? That is one way to reduce our healthcare costs. Right? So it's a big challenge, but to meet that challenge, we really have to understand the molecular basis of human variation. So the first thing I want to talk about is, well, what do we know about it? What is the nature of the variation between us? And this graph sort of, it outlines three general types of variation that we know about and something about their frequency and how big they are. And as you might imagine, the things that are the most common are also the smallest. And those are called single nucleotide polymorphisms. So in the vernacular of Bill Clinton, who called the DNA sequence of the human genome to be the book of life, in the book of life, a single nucleotide polymorphism is a single change in a letter of that book of life. A replacement of one for another, let's say. Now, there's another type of change that we've known about for a long time, of course, which is changes in whole chromosome numbers. Trisomies or monosomies, that is, loss of a whole chromosome, or gain, the duplication of an entire chromosome. And the other type of change, which is more recently appreciated, is this one in the middle, copy number variant. And in the vernacular of the Book of Life, again, these are not individual letters of that book, but they're words, or paragraphs, or even whole sections of chapters that are deleted or duplicated or transposed somewhere else into the text of the Book of Life. Now, all of these, it's important to bear in mind that these different types of variants are measured in different ways. We are always affected by the nature of the assays we employ to understand the differences between us. And the truth is that the majority of the effort we've put so far in understanding human variation has been on SNPs. And in part that's because there was great technology available through commercial enterprises to study that type of variation. But as it turns out, the copy number variation, the deletions or duplications, those are measured in somewhat different ways. And our appreciation of those variants has been fairly recent. So let me talk a little bit about what they are. First of all, we've all got them. that was one of the first surprises. When we started looking at this type of variation in, quote-unquote normals, we found that all of us have this type of variation. It is not unique to disease at all. It's found in all of us, and there is a lot of it. So these are deletions and duplications of varying sizes, from hundreds of base pairs to millions of base pairs. The other thing that we came to understand is that this type of variation, it's not randomly or evenly distributed in our genome. Quite to the contrary. There are places in the genome which are highly variant. And these regions occur in parts of the genome where there are big, highly related, duplicated sequences. Okay? And these are sequences that are not duplicated thousands or hundreds of thousands of times. These are sequences that are duplicated 10 times per genome, 14 times per genome. And so one of the terms for them was called long LCRs, or long complex repeats. They're also known as segmental duplications, but their characteristics are that they're big, they're relatively low copy, that is 10 to 14 sequences, and they're highly related to one another. That is 90% identity between one related sequence and another. So I want to show you the distribution of these creatures in the human genome. So here's the entire human genome, separated by chromosome. And these red hatches you see, these blue connectors, are connecting related sequences on the same chromosome. Okay? So all those arrows, if you will, those V's, represent the places where these sequences are related to one another on the same chromosome. So there's a lot of them. Okay? Now, there are about 170 or so regions like this in the human genome, and a very high number of them, changes in those regions are associated with human disease. Okay? So they're clearly playing a role in human disease. Now, they play a role in evolution as well. So I want to show you the distribution of this type of sequence in two species, the human and the baboon. So this is a depiction of chromosome 16, and related sequences are color-coded. Okay? And you can see that the baboon chromosome 16 on the right and the human chromosome 16 on the left, they have a very different representation of these sequences. There's a lot more of these duplicated elements in the human genome compared to baboon. And in fact, this group of authors feel that the bursts of variation, of speciation in the hominid line, have occurred when there were bursts of these duplicated sequences appearing in the genome. So they postulate that the connection between our evolution and the connection between our genome change is very close indeed. So let me show you a little bit of another species, the mouse. And it really, I think, highlights the differences in complexity of this type of sequence between different species. On the left are humans, and on the right, the mouse. And in this case, the blue things that you see connect the sequences related on the same chromosome, and the red lines connect them between different chromosomes. And the top two graphs are these sequences that are only bigger than 10,000 base pairs, or sequences that are bigger than 20,000 base pairs below that. So you can see that there is a big difference in the complexity of these sequences in these species. And you know something? There's a big difference in the stability of these species. The mouse has been around for a long time, doing quite well, thank you very much, and it hasn't changed all that much. And there are other species we know that's also true. Sharks, they're pretty successful. And they haven't changed that much for a long, long time. And they're probably going to be here a long, long time after we are history. Right? So not all species change at the same rate, and neither do their genomes. And of course, we are the wacko-lunatic species. We are unstable in our behavior as well as our genome. Okay, so I want to get back to this. What is the consequence of this type of organization? And the consequences are that there are disorders that we now call genomic disorders that really are a consequence of rearrangements in those intervals of the genome. Deletions, duplications, inversions, and translocations. And again, these rearrangements are associated with these sequence architectural features. Segmental duplications are also known as low copy repeats. All right, but what I want to tell you a little bit today is the application of this understanding of our genome to understanding this disorder. So what is this? Well, it's a behavioral disorder of children, and its hallmark characteristics are language deficits, repetitive and stereotyped behaviors, and altered social engagement and interactions. Right now, its incidence is measured by the CDC is about 1 in 88. 1 in 88. That's a big number. Just by comparison, childhood cancers, 1 in 800. Okay, so this 1 in 88 number means the impact on our educational system, on our medical system is huge. So there is some good reason to understand this. Now, what do we know about the genetics of it so far? Well, first of all, it's called autism spectrum disorders in most circles, and that reflects the variation of the symptoms that these children exhibit. It is very broad, and it's found with other disorders. So these characteristics, they're not just unique and stand alone, they're found with other characteristics as well, with other disorders. That one type of variation that I talked about, SNPs, we really haven't found any of that type of variation at all really associated with autism in any significant number, but we have found a lot of those copy number variants. If we do a pretty good assay on any child with autism right now, we can identify an event, a deletion or duplication in that child, that is probably responsible for it in about 5 to 10% of the cases. So we know that type of variation is indeed pretty important. But of course, it also points out that since this is true, and all of these variants, I should say, they're in different regions of the genome. So it's not like there are 10 genes here. There are probably hundreds to thousands of genes that each contribute in some way to autism. And it also points out that even with this set of events we've been able to discover, the heritability of autism is about 80%. That is, for example, if you have one identical twin and another, if one of them has autism, the chances are the identical twin has one is about 80%. So where is this genetic variation? The answer is it's still undiscovered. Okay, so here are the questions that we began this study that I'm going to tell you about very briefly in 2 minutes and 51 seconds. What is the total amount of copy number change associated with autism? And if we find it, what is its nature? And where is it? Is it duplications? Is it deletions? Is it randomly distributed through the genome? And is there a relationship between how much of this genetic variation occurs and how severe the phenotypes are of these children? Is there a linear or correlative relationship there? So here's the first piece of data that I'm going to show you. And this represents the level of copy number change by simply saying, let's plot the largest copy number variant in that individual. And then that's on the x-axis, and on the y-axis we'll plot its frequency. Now here are the different groups. This top group, which has the most copy number variation, right? That group is a group with actually severe intellectual disability. They have the highest degree of copy number burden, if you will, copy number change. These two in the middle, the purple and green lines, are two different sets of children with autism, totally evaluated by two different groups entirely independently, showing the same and an intermediate level of copy number variation. And the last two lines are the controls. So what this shows is there's a gradation of copy number change. And I'm going to show you to another way where we're doing the simplest kind of analysis. We're simply adding up the total number of base pairs of duplication or deletion in these children with autism. And what you see is on the right-hand graph, which is one set of children with autism, that there's more duplication. There is no difference in the level of deletion. And the same is true for the other cohort shown on the left. So, is there a correlation between how much duplication burden there is and how severe the phenotypes are. And the answer is there is if you measure it in the right way. And this is a measure, the Vineland score is actually an interview of the caregivers of these children that looks at 4 different areas: socialization, communication, daily living skills, motor skills. And not surprisingly, where we see a significant correlation between the amount of copy number change and these measures of clinical outcome is for socialization and communication skills. Okay. So, in the last few slides, I'm just going to show you where is this burden. I'm going to summarize this for you. The burden, this duplication burden, it's in the neighborhood of those segmental duplications. It's not in unique sequence throughout the genome. It's in the neighborhood of those segmental duplications. So those crucibles of change, that's where this duplication burden is. And we're comparing blue to red as autism and controls, and you can see there's no difference in the deletions. Those lines match, but not so in the duplications. Okay. I'm going to get down to the take-homes here. First of all, these neurodevelopmental disorders are associated with total levels of copy number change. Autism is a disorder, surprisingly, of duplication. And you might think, why is that? Deletions, which is going to be more deleterious, a deletion or a duplication? Likely when you pull function out, right, with a deletion. And that variation occurs in those crucibles of change. Okay, here's my last slide. What I've told you is that autism is a disorder of duplications, and what I didn't have time to show you, one of those slides I moved over, was that more severe disorders have both deletions and duplications. Okay? But what I haven't talked about, which I think is very important, and actually harkens back to some of the things that Dr. Hudson said, is that, yes, when we do these experiments, we're looking at the selection, but isn't it possible that the environment might influence this? What if the environment changes the stability of these hotspots of change in the genome? That is a mechanism for the environment to influence a genetic disease. And there's a big debate in the community. Is autism incidence on the rise? It depends who you talk to. But this is one mechanism where environmental factors could influence it. And we know of environmental factors that influence genome stability in exactly these parts of the genome. So this is something that we should be concerned about and we should be measuring. It wouldn't be appropriate without some acknowledgments. This is a team of many from many institutions. It's very common in science these days that if you're going to approach these problems, you reach out across institutions and different types of fields. And these are some of my colleagues at UC Davis at the Mind Institute there. Some of them are here. That's a picture of me when I was 12. And I'm happy to say that some of these folks who were at the University of Washington, we have recruited to Penn State University, and they're working with us here. Anyway, my thanks.